Welcome to our AP Biology video. Today we will be explaining the process of DNA replication. DNA replication is the process by which DNA is copied in the S phase of interphase before the cell divides to ensure that each daughter cell receives the correct amount of chromosomes. DNA, or deoxyribonucleic acid, is a nucleic acid made up of three components: a 5-carbon deoxyribose sugar, a phosphate, and a nitrogenous base. The structure of DNA is a double helix. DNA has 4 bases: adenine, thymine, cytosine, and guanine, which form pairs. Adenine pairs with thymine, and cytosine pairs with guanine. Adenine and guanine are purines, meaning that they have 2 rings, while cytosine and thymine are pyrimidines, meaning they only have 1 ring. The first step in DNA replication is the formation of the replication fork. The enzyme helicase disrupts the hydrogen bonds between the base pairs of the DNA and separates the strands into a Y-shaped replication fork, which is where the template for replication will begin. DNA can be labeled with a 5' and a 3' end. The 5' end has a phosphate group attached, while the 3' end has a hydroxyl group attached. Replication only progresses in the 5' to 3' direction. But the replication fork is bidirectional, meaning that one strand is oriented in the 3' to 5' direction, called the leading strand, while the other strand is oriented in the 5' to 3' direction, which is called the lagging strand. Because of the directional difference, the 2 sides are replicated with 2 different processes. The second step in DNA replication is the primer binding. Once the DNA strands separate, a short piece of RNA called a primer binds to the 3' end of the leading strand. The primer binds as the starting point for DNA synthesis. Primers are generated by the enzyme DNA primase. Step 3 is elongation. DNA polymerases create a new strand through elongation, where the DNA polymerase binds to the strand at the site of the primer and starts to add new base pairs complementary to the strand. Since replication proceeds in the 5' to 3' direction on the leading strand, the newly formed strand is continuous. The lagging strand binds with multiple primers, which are only several bases apart, and begins replication. DNA polymerases add pieces of DNA called Okazaki fragments to the strand between primers. These fragments dedicate synthesis away from the direction of the replication fork. The newly created fragments are disjointed, causing this process of replication to be discontinuous. Step 4 of replication is termination. Now the continuous and discontinuous strands are formed, and an enzyme called exonuclease removes the RNA primers, which are then replaced with appropriate bases. Another exonuclease checks the new DNA to remove and replace any errors. This enzyme removes successive nucleotides from the end of a polynucleotide molecule. Another enzyme, DNA ligase, joins Okazaki fragments to create a single unified strand. A section at the end of the parent strands consists of repeated DNA sequences called telomeres, which act as protective caps preventing nearby chromosomes from fusing. The telomere in humans consists of the bases TTAAGGG, which is usually 3,000 to 15,000 base pairs long, and every time DNA is replicated, 25 to 200 bases are lost. An enzyme called telomerase catalyzes the synthesis of telomere sequences. Lastly, the parent strand and its complementary DNA strand coils into a double helix shape, and these can further condense to form chromatin, which is a tightly packed and wound DNA. Thank you for watching. This has been DNA replication.